Could you please introduce yourself and tell me what course you teach?
Yeah, my name is Jack Ikeda, and I teach MCB 428, Microbial Pathogens Laboratory. And I got my PhD in microbiology at UC Davis in California, and I first taught this class in 2011. It's an advanced MCB lab course that fulfills MCB major requirement.
When is your course typically offered? In the fall, spring, or summer?
The course is typically offered in the fall.
Can you give me a brief overview of what is covered in your course and what are the learning goals?
So the course is like a diagnostic lab where you're given clinical samples and you try to identify the pathogen in those samples. Basically the learning goals is learning how to work with microorganisms in the lab using good aseptic technique, and biosafety is always a major concern in the class, so you learn about that. And you also learn about the pathogens and how they can cause disease and spread.
What is the format of your course? How often does it meet?
It meets twice a week, 2 hours each session. It's a lab course.
How are students in your course graded? Exams, quizzes, or homework?
So the grade is based on 2 exams and a final exam. There's a lab practical, and there's 7 exercises that you have to write lab reports for. Each one has a pre-lab and a post-lab that you have to do online Kappa. And there's 100 points possible.
Historically, which portion of the grade breakdown do students struggle most with?
I would say the exams and maybe the post-lab questions, because they're short answer questions that make you think. The post-lab questions, though, you have a week to answer. But so the exams would probably be the most difficult part.
Are there points associated with attendance?
Being a lab course, it is mandatory that you attend. You are allowed to excused absences, but after that, you might be deducted points because each exercise has so many points and so many days associated with that exercise, and so each day has a certain number of points. And so if you have an unexcused absence for one of those days, you would be deducted those points.
Are there any required textbooks for this course?
You have to buy a lab manual, the current one. You can't use an old one because it's constantly changing.
What kind of background knowledge do I need to take this course? Are there prerequisites?
There are prerequisites, and as a professor, I always strongly recommend that students follow prerequisites. They're put there for a reason. You know, the professor wants you to have a certain knowledge before you come into the class. And for this class, MCB 300 is a prerequisite, microbiology. And I expect students coming in to know basic microbiology, what's a bacteria, what's the difference between a gram-positive and gram-negative organism, how they grow. People who haven't taken the prerequisites, they might have to work harder to understand the material. There's a lot of terminology associated with any science, and that's how I describe these things. And so I'll be using terminology hopefully that they already learned in 300.
How do topics covered in your course apply to current research or medical and scientific advancements?
So topics that we cover and techniques are the basis for a lot of the modern diagnostic techniques used today. Today, a lot of the diagnostic tests have been automated, and they use molecular techniques looking at DNA. But the old classical approach, staining and microscopy and biochemical tests are still important, and using antibodies. And so you need to know those basic techniques too.
What opportunities do you provide outside of class for students to ask questions or get assistance?
So I have office hours twice a week, and each of the TAs has office hours once a week. A student could go to either of those, their office hours. You could email any of us questions, and if you can't make office hours, you can always email us to see if we can meet at another time.
What is your favorite experiment that the students perform?
I would say the gastrointestinal tract infections lab, just because it involves one of my favorite organisms, Salmonella, because I used to work with Salmonella as a graduate student and a postdoc. So I would probably say the GI tract infections lab.
How helpful do you feel this lab material is in helping students understand other courses in the MCB curriculum?
Well, a lot of the other MCB advanced courses are related to microbiology. So taking a microbiology lab course and working with actual microbes, I think, gives you a greater experience and hands-on, and you kind of get to know these microbes better if you worked with them. And so I think it would help, especially in those microbiology courses.
Are there lab practicals in this course? If so, what is the format?
Yeah, there's one lab practical at the end of the course, and we test the students individually on streaking plates and gram staining and using the microscope and scoring biochemical tests. It takes place over 2 days, and there's stations that the students are at, and you're timed, and then they switch to the next station.
How would you describe the workload for this course? Is there an estimate for how much time students will spend outside of the class?
Well, it's a 2 credit hour course, so it's less than a 3 credit hour course. But the students do have to come prepared to the lab. There's 7 exercises, and so there's 7 pre-labs that they have to prepare for. They should read the lab manual before coming to class because they need to know what they're going to be doing that day. So there is quite a lot of work, like pre-labs, lab reports, and post-labs that are constantly due. You need to keep up with the work.
What techniques taught in this lab can be transferred to other parts of research?
Well, just being in a lab and learning about lab safety, I think, is pretty important, especially if you're working with microbes. You have to learn about biosafety, and so that's definitely something that you would get out of this class, and learning just basic microbiological techniques and aseptic technique is pretty important so that you don't contaminate yourself and others and your sample. If you've never been in a microbiology lab, that's something that you have to get used to, the aseptic technique part. Not putting, leaving plates, lids open so that microbes can get on the media, and putting down the sterile wooden sticks on the lab bench and contaminating it.
Is there anything else that you would like to tell us about your course?
Well, if you're interested in microbiology, this is one of the 2 microbiology lab courses that you can take, and this one involves pathogens. Microbes are everywhere. Some of them cause disease, but a lot of them are good. Most of them are good, and we need them, and so microbes are everywhere. I think everybody likes microbes, and you get to work with microbes.
Okay. Thank you.
You're welcome.